Good morning, everybody. I'm Ushasri, working as Assistant Professor in the Department of Biomedical Engineering. Today, we'll be continuing the topic of biocompatibility. In the previous class, we have discussed about various materials and their level of biocompatibility and how important it is. So, today, we'll be looking into the testing of biocompatibility. So, as discussed, it is a part of course name, that is biomaterials. And these are the course objectives and course outcomes. In today's class, we will be discussing about the definition of biocompatibility and what are the various requirements of biocompatibility, testing of biocompatibility, what are the different levels of testing which we have in order to evaluate biocompatibility, and also the applications where we use these testing methodologies. So coming to the definition, biocompatibility is defined as the interaction between the biological tissue and the biomaterial that is being in harmonious with the tissue host environment and having no toxic or injurious effect on the biological functions. So when a material is implanted inside the body and it is coming in contact with the tissue, the interaction between the biological tissue as well as the biomaterial is of major importance in order to assess whether it is compatible enough or not. To do so, we will be studying both the aspects and both the interactions on to the implant material part as well as the biological tissue part. The ability of the material to perform with an appropriate biological response is the major intended application of this analysis. So, if we are able to assess the interaction between the biological tissue and biomaterial is safe enough and it is not causing any adverse effect on the tissue, then it is feasible to continue to implant the material inside the body and ensure that it is performing well without causing any hazardous effect to the tissue. This is what about biocompatibility. So coming to the requirements, as discussed earlier, biocompatibility is the interaction between biological tissue and biomaterial. So here the biological tissue is of major importance, wherein if the introduction of the material inside the tissue causes any change in its cell metabolism, function, in its shape, size, survival, any aspect, it is of major importance to assess if it is causing any damage, to what extent, what type of damage is caused to the tissue. So in order to have the requirements, or in order to discuss the various requirements of biocompatibility, we have to assure that the tissue material and the biomaterial both are in friendly environment, and they are and it is very feasible to work with the material for the given application. So for this, it should not, the first and foremost major requirement is it should not cause any harm to the living tissue inside the body which is coming in contact with the material. Next, it should not elicit or create any reaction onto the host tissue which causes some irritation, swelling, inflammation, or any such allergic reactions which may further lead to allergic reactions for the patient. And if the material is implanted inside the body and is supposed to remain inside the body for the entire life of the patient, the another important aspect which we look into is its performance, reliable performance throughout the life of the patient. So soon after the implantation, it may not cause any harm to the tissue and may not cause any allergic reactions also. But the prolonged use of such materials for the entire life of the patient may cause some elements of the biomaterial which are getting degraded into the biological tissue and are getting carried through the circulatory as well as the lymphatic system. In such cases, the local response, that is the response of the material soon after the implantation, may be compatible enough, but prolonged use of such materials may lead to systemic response, that is wherein the elements of the material may get leached into the other systems and affect the system function on the whole. So here when we want to discuss the performance of a biomaterial for a given application, we would be majorly looking at its response at the tissue material interface. So after implanting the material, when the material is coming in contact with the tissue, the interactions occurs on the both sides. That is, the material will be getting affected because of the physical contact of the tissue environment, and as well as because of the material, which is a foreign body for the tissue environment, it also undergo few changes, and there occurs a cell reactions onto the biological tissue part. So the tissue material interface is the major area which we need to study in order to say whether it is compatible enough to work with that material to the extent wherein it is not causing any harmful effect to the tissue. For this reason, we study the biological response of the biomaterial in terms of two aspects. That is the implant material which will be used as a biomaterial, and host response, that is the response of the living tissue when the material is coming in contact with it. Once we are clear with the requirements, what exactly need to be studied, prior applying that into the human environment, we need to test for its compatibility. From the preliminary studies, we will be able to assess what are the materials suited or biocompatible with respect to the living tissue. For example, if we want to take any orthopedic implant material wherein we want to replace that for fracture healing, a bone got fractured and a material need to be replaced in order to augment the process of healing. The fractured bones are misaligned, so first and foremost we need to align them by using some external fixative devices such as braces, pins, screws, etc. And then we need to ensure that the tissue is healed. If it is a multiple fracture wherein healing takes a lot of time, then we will be using plates, bone plates also to assist the healing. So in doing so, the tissue is coming in contact with the bone tissue, surrounding muscle tissue and all those things. So here, if we want to test that material is perfectly working for that healing process or not, we need to priorly assess that this material is suitable to work with respect to the bone tissue, muscle tissue, which is coming in contact with the material. So such studies need to be done prior implantation, and then after assurance of their compatibility with the host tissue, only then they will be implanted into the body. If you want to analyze the biocompatibility and its level of application with respect to a given criteria, first we need to test the performance of the implant material, and there are some standard organizations such as ANSI and ISO which would be giving various protocols for testing, the preliminary studies, then the animal studies, and then the usage test wherein we will be including the clinical trials. So prior application into the human tissue, first we go with the laboratory test, that is we are performing the tissue material interaction analysis outside the human body. So hence these tests are called as level 1 test, which are in vitro. In vitro in the sense outside the body. Next, after assuring with the results that it is compatible enough to use this material for the given application, then we will check with the living tissue. That is, we try to implant the material inside a species, that is an animal species, which has the match with respect to that of the human tissue. So once we selectively choose a particular animal species and implant the material inside the animal, then we check for the biocompatibility levels by placing the material inside the body, and hence the tissue material interactions are studied in vivo. In vivo is inside the body. So to perform this in vitro and in vivo analysis, there are standard protocols given by the standard regulations such as ANSI and ISO, following which we can assess that based upon the results of the preliminary test, that is the in vitro analysis, what type of protocol to be followed in vivo analysis. So that is how we categorize the evaluation techniques as level 1, level 2 and level 3. In level 1, which are in vitro, that is outside the body, in the laboratory we perform this analysis and then perform the preliminary tests. Evaluating the tissue response outside the body would be would be done based upon few assumptions. So in order to have a furthermore clarity on the test results, then we will be going with the in vivo test, wherein we perform the analysis inside the body, and here the body would be of a selectively chosen animal species. So hence these are also called as animal test or the secondary test. In the level 3, wherein we have successful results with respect to level 1 and level 2, then we can assure that it is safe to implant the material to test the biocompatibility inside the human body. Then we selectively choose the human volunteers or the cadavers, which will be used for testing purpose. And in this, we try to implant the material as intended for the application in the host environment, exactly similar, the same way how it is done with respect to patient. Then we will be having a complete picture of the biocompatibility, its severity, its tissue material interactions, and the applications which can be possible with the level of to what level of biocompatibility. So these are done in animals which are of sophisticated when compared to the animal test like monkeys, and which have close match with that of the human species. And in some cases we can also go with the human volunteers as well as the cadavers. The other name given for this is the usage test or the clinical trials. So having understood the various requirements and various types of testing methodologies for biocompatibility, now we try to understand what are the different aspects in each level of testing. So in level 1, that is the in vitro test, we are mainly interested in cell metabolism. So here we mimic the host tissue cells with respect to the tissue material interaction outside the body in a laboratory, for which we use various cell culturing medium in test tubes, culture plates and various other modalities. Using these modalities in the laboratory, we try to analyze what exactly happens to the cell when the implant material is coming in contact with that tissue. So here with the level at cellular level, we will be able to assess whether it is safe for the tissue or not, for which we will be checking for the preliminary study of cytotoxicity. The cytotoxicity test would be of major interest because it will be giving a clear-cut idea of whether the material is safe for the cell or not. After implantation, is the cell being damaged or killed or being reacted in any other form which is not desired for its metabolism? And then if in case it is killing the cell, then there itself the material will be not promoted for the next level test. And this level of testing we call it as cytotoxicity. Here we get an idea whether this material is toxic to the cell or not. So that is about the cytotoxicity. Next is the cell metabolism with respect to mutagenic effect. Each cell has its own dedicated function to perform. If the implantation of the material is going to cause any change in the cell metabolism, wherein its function is being altered, modified, or in any way going to damage the cell and being affecting the surrounding tissue, then that is of not required with respect to the material. So if a material is causing any such reactions with respect to the cell metabolism, then we will be not selecting that material and it will not be a biocompatible material for that application. Metabolism would be checked with respect to its function. So even this analysis can also be done in the in vitro analysis by taking few allergic sensitization and membrane test. So wherein the particle, how it is being taken by the cell, whether it is being accepted by the cell or not. If it is not accepted, in what way the cell is going to react towards that material. Is it discarding it, rejecting it, or is it accepting it and converting it into a form which is acceptable to the tissue? How the material as well as the tissue is going to get modified after the material is implanted. All such analysis and information would be known from this test. And the third one is for the systemic response, that is mutagenic test. In this mutagenic test, the prolonged application of the material, is it going to cause any carcinogenic effect or not? Cell mutation wise, first in toxicity we have identified whether it is toxic to the cell or not. In the metabolism test, we have identified whether it is going to alter the cell metabolism in any form or not. Now, is it going to change the cell itself as such? The inherent property of each cell is defined by its DNA. So, if the DNA is getting altered or changed, then on the whole the cell entire, its metabolism, structure, shape, form, everything gets changed. So is there, because of the prolonged application of the material, is there any change in the mutagenic reaction of this DNA with respect to that cell or not? That will be analyzed. So the results of this mutagenic test would be giving a clear-cut idea of whether the material is going to cause any type of toxicity or carcinogenic agents on the prolonged application. So if, say, suppose the material is inside the body and it needs to be there for the entire life of the patient, what is the systemic response? Local responses, it is fine, it is compatible enough, it is performing well and well accepted. But because of its prolonged application, is there any material getting leached into the circulatory system, or is there any material property which is getting altered, or is there any mutagenic change within the cell which is causing the entire metabolism of the cell? All such things will be there. So usually the level of carcinogenicity is cancer causing elements. In most of the cases, what happened, because of this prolonged application of the material inside the body for years together, these material particles tend to degrade and then try to enter into the other circulatory systems or the lymphatic network which is connected to that tissue. So when this happens, the cell gets changed mutationally, and then its property will be changing and it tries to get proliferated multiple times, and this uncontrolled undesirable proliferation of that tissue may cause cancer. So that is the level of carcinogenicity which can be assessed with respect to mutagenic test. So having performed the level 1 test outside the human body, now we further move to the level 2, which is in vivo testing analysis. In vivo testing analysis, preliminarily prior going for usage test, which will be including humans, we will be interested in testing in animals. So in order to test the performance of the material inside the animal, we have to choose the animal species, and this animal species is selectively chosen based upon the close match which it gives with respect to human tissue. So suppose for heart valves, if I want to check an artificial heart valve and its performance in the heart, so the valve size, shape and the cusp, everything should have a close match with that of the human tissue. So why we selectively choose cow and pig for the heart valve analysis is like the size, shape and the texture and the functioning of the heart valves are having close match with that of the human valves. So having such data, we will be selectively choosing a particular animal for the analysis, and then we go for all these types of tests. Again in this, the membrane test would be giving like whether it is being identified as a friend or foe for the tissue. If it is being identified as a foe, what is that form in which it will be rejecting that material? Is it discarding the material right away by causing any inflammation, infection, or any such thing? Or is the material getting leached away and getting into the circulatory system by causing any systemic response? All such information will be given. Skin sensitization test, few of the materials would be causing some irritation and skin sensitization, wherein we can see redness, swelling on the skin because of the material interaction. So such test also can be analyzed by this in vivo application. And systemic toxicity is like in the in vitro we have discussed about cell toxicity, and here we will be able to assess the systemic toxicity, that on the whole the tissue, is it going to get toxic, is it going to cause any cancerous tissue there or not. Such details will be given. And coming to the usage test, here I am explaining with respect to a dental application. So the material after being tested in the animals, if it gives successful results, then based upon those results, the same implant material with the respect to the size, shape, form, and all those considerations would be implanted into the human tissue. And say suppose for dental application, we want to replace the artificial tooth. The tooth is missed or dislodged, so we want to replace it with an artificial tooth. So in that we have to fabricate the artificial tooth with an artificial material which need to be implanted into the gums or the root where we want to exactly situate that. So it will be getting in contact with the gums and the pulp which would be holding that artificial implant into the gums. So first we have to check whether that implant material, since it is coming immediate contact with respect to artificial tooth, is your gums. So the gums are getting infected or not, whether it is able to perfectly get fixed into the pulp area in the gums, which would be anchored and have a grip, give a grip to the artificial tooth or not. So then we will be going for the pulp capping and all those things. Like we want to have a cap on that artificial tooth. We don't want that material to be looked as an artificial one. So we will be giving a ceramic cap onto that. And whether this cap is feasible or not, like whether it is giving the natural look such when compared to the other tooth or not, and then whether it is fixed onto the artificial tooth and get anchored into the gums perfectly or not. Otherwise it would be just getting dislodged and all those things. And the endodontic usage test is like onto the final end after implantation and all those things, if the patient is able to sustain it for his natural requirements. Like tooth, when mastication, it should not get dislodged. It should provide a ease of mastication for the patient. While chewing, it should not cause any pain to the patient, nor it should get loosened, nor it should not cause any bleeding in the gums and all those things. So that would be the final end usage test, which would be finally giving a result based upon which we can decide whether this material can be used for designing artificial tooth or not. So these are the 3 levels of test. Now going a bit in detail with the testing methodologies. In vitro, as I told you, it is the preliminary test and it is performed outside the body, and hence we perform the, or we try to simulate the tissue material interface in the laboratory by using various test tubes, cell culture dishes, and outside mediums wherein we use some microorganisms also in order to culture the tissue medium inside the culture plates. So what are the major advantages? First and foremost thing, we are performing it outside the body. Hence, it is not causing any sort of damage, or it is not involving the human tissue at all. So the patient is always on the safe side, and he can assure that if it is like perfectly okay with respect to in vitro analysis, then it is completely safe to get it for the next level of test. So it will be, since we are performing in the laboratory equipment, we will be readily observing the results, and then it will be very quick to assess whether it is compatible enough or not. And being it in a laboratory equipment wherein we are not including any human volunteer or any animal species for testing, it is of less expensive when compared to the usage and animal test. And these tests are the preliminary test, and the results obtained from these tests would be of major interest, and they will be setting the standard to standard such that the animal test would be giving furthermore information in addition to these results. Well suited for large scale screening and good experimental control, because since everything is under control of the man who is performing the test, so can be tightly controlled and address specific questions which would be of scientific approach, like how far it is compatible enough or not. If compatible, what is the level of interaction, what is the type of interaction, how far it is safe to go with the animal test and usage test. So all these technical details will be clearly understood by using such test. So here, like major criteria, as I told you, would be of cytotoxicity. So here, as I told you, whether it is causing any toxic effect. This may be in terms of cell membrane permeability, whether any abnormal transport is going to occur at the cell membrane, because each cell is surrounded by its membrane, and that is the wall which protects the cell internal structure. It will not allow any foreign body to allow into the cell. And if in case any foreign body enters it, first it will try to understand whether it is a friend or foe to the cell. If it is a friend, any protein molecule, any biological molecules which are used for the cell metabolism, yes, it will be readily accepting it and then making use of it. In case if it identifies as a foreign material which is not at all useful for the cell, then it try to encapsulate it. Your lysosomes present in the cell would be encapsulating the foreign material and try to push that out of the cell by means of exocytosis, and then it will be carried into the lymphatic system as an excreted material and will be discarded outside the body. So here the membrane test and the metabolism test would be of major interest. Toxic enough or not? If toxic, what are the changes which are going to occur? So this would be of major interest for cytotoxicity. So here, whether it is causing cell lysis or not. In few of the material interactions, what happens is soon after the implantation, these materials try to kill the cell because they are not at all feasible. They will be turning into very toxic for the cell, and the cell get killed, and that is called as autolysis. By its response, the cell may accept or reject the body, as I told you. If it is accepted, yes, it will accept it, and if it is rejected, it will be sending out through exocytosis. This acceptance or rejection would be in the tissues, organs, and whole body is made up of whether the foreign material is compatible enough or not. So here this will be giving a level of understanding on what is the level of biocompatibility with respect to this material at the local host tissue level as well as at the systemic response. That is the surrounding tissue and all the other systems which are getting involved with that environment. So as I told you, coming to the methodology, we will be majorly focusing on the cell culture, because we try to mimic the cells which are getting affected by that implantation in the laboratory equipment, and then we introduce the material into that cell and then study the tissue material interactions. So for this, we will be using cell culture. It may be a primary cell culture using cell lines, wherein we try to form the cell lines onto the culturing medium and then we test its performance individually. Or it may be a continuous cell line, wherein we have culture the medium and we place the material and try to take the information for longer duration. So both local and systemic response both can be assessed by using such methodologies. So this is one example. Like here, this is the test tube wherein you have a culturing medium into which we have cultured the cells, and this red one which would be of major interest because that is being getting altered because of the implant material, and that would be identified at various levels. So here in the first thing, they are treated with the enzymes and try to identify which cells are getting affected, which cells are not getting affected. Once they identify the cells which are getting affected, they try to separate it out in the culture medium and then try to see what type of changes are going to occur. So in the doing like continuous cell lines are used, then the cells which are not getting reacted get added at one side, and the cells which are getting manipulated and resulting in the proliferation, abnormal proliferation can be easily identified. So this is how we can check for the cell toxicity using cell cultures. And next one is the membrane permeability test. As I told you, that membrane is the one which identifies what is required for the cell and what is not required for the cell. So here the advantages are it identifies cells that are alive and dead under the microscope, and it is important to identify the cells that are physically present but dead. So after the implantation, when the material try to interact with the cells, whether it is causing any autolysis or not, meaning that is it killing the cell or not. Few cells will be killed, few cells will be damaged, few cells will be getting unaltered, like we have seen it previously in the test tube and culture mediums. So like it will be feasible enough to perform the membrane test to identify what are the cells getting affected, and if getting affected, in what form they are getting manipulated. That would be of major interest. So disadvantages with this are the questionable relevance to the final in vivo application. So it is done externally. We know that it is toxic, but to what level? Is it safe for human application or not? Usage test or not? Is it safe for the animal test or not? So because though we are selecting an animal species for analysis, even it is a living species, and we cannot sacrifice the species just for the purpose of testing the material response. So hence, prior implanting the material into the animal and perform animal test, we should ensure that it is not threatening its life. It is safe to use with respect to that animal. It will not cause or kill the animal in the process. That assurance need to be taken. Only then ethical permissions will be given, and then animal tests are performed. So like as I told you, cell metabolism wise, like metabolism, each cell has its own metabolism, a particular dedicated function to perform, and this will be decided by its DNA and its cell internal organelles. So the level of biosynthetic or the enzymatic activities that occur in the cells would be of major interest to assess the cell toxicity here. So the synthesis of DNA and protein is analyzed by the adenine radioisotope. Like we try to label the each cell with respect to some standard control, which we know, like for that material, what type of reaction would be given by that material. So making use of such labels, we try to set the standard control, and with respect to that standard control, we try to identify the other materials and the response of the host tissue as well. So these are few culturing mediums shown here, in which we are testing for the cell metabolism, and we can see like we have different shades of the agent. Once that agent is here, MTT is one such agent which is being given to that. If it is going to react, then it will change the color into dark, or else it will be not resulting any change in color. And if it is there any slight interaction not that toxic to the cell, then the shade of the color change would be of a different contour. Based upon the color change, we will be now able to assess whether it is toxic to the cell or not toxic. If toxic, to what level. So this MTT is like an agent or a standard which is used to analyze the cell metabolism changes because of the implantation material. So then this is a spectrophotometer, a device analytical instrument which is used to assess the concentration of the material and its interaction with respect to the tissue. If it is causing any change, toxic effect to the tissue, so what is that, in what form it is, how much of severity is there with respect to that toxic, everything can be analyzed by using this machine. So these are the mutagenic assays, like in the DNA, like when we use this, so they will directly, if the material is going to directly alter the DNA or not, or is it requiring any activation, transformation of the mutagens at the DNA level. If it is going to change the cell characteristics, then it would be of major concern in terms of biocompatibility. So for those, we will be using genotoxic mutagen, epigenetic mutagen, like various other level, hormonal level, mutational level, DNA, protein synthesis level. All these are the inherent characteristics of each cell. So at that level, if it is, the implant material is going to cause any change, then there is a chance that it may turn into carcinogenic. So that is a clear indication. Like if these test results of mutagenic assays are giving a positive result, then it indicates that there is a chance of carcinogenicity. Ames test is also the same test, most widely used test in order to assess for the mutagenic interactions. And in this, we use a like how we were using standard in the previous membrane sensitization test. Like here also we use a standard which would be used to identify the change in the cell mutations and then try to identify whether it is carcinogenic or not. So like this is salmonella is one standard which is used to identify the mutagenic reaction with respect to liver extract. So here the sample is the host tissue is liver tissue and that tissue will be used. So in the experimental test tube, we will be using suspected mutagen, like what is the suspected mutagenic reaction going to take place, and then we mix it with the standard, that is the salmonella, and then we see like where we have only control with liver extract and there is no mutagen being added, we don't get any abnormal tissue growth. But in the culture plate where we have added suspected mutagen, like is there any chance to change this, or is there any chance for the cell to get this mutational change or not? If we suspect a particular thing and make use of few elements, agents, mutagenic agents, which would be added to that. If it is getting reactive, yes, it will be indicated in the cell culture plate, and that is how we can assess whether it is carcinogenic or not. Coming to the in vivo test, these are the secondary tests and called as animal test, where we choose a particular animal species with a close match with that of the human tissue and then check for the tissue material interactions. So usually rats are the common animal species which are used for various sensitization tests, and the response of these animal tests are more comprehensive, they are relevant in vitro, and these features are major advantages because prior human applications we are going for this animal test. So though we claim it to be in vivo application, the test results always depend upon the type of the species selected, and the reactions given in the animal test would be having a close match with that of the human behavior also. So here the advantages with this test is tests are done for irritation, allergic, systemic toxicity as well as cytotoxicity. and any local reaction at that site with respect to the contact of the material, like at the tissue material interactions, is there any irritation, sensitization, any sort of reddishness, or is it causing any skin irritation, skin allergies? All these things will be analyzed. The local reaction is an inflammatory one with the redness, itching, and the reaction which will disappear if the material is removed. Like soon after the removal of the material, again it is coming back. So it is a clear indication that yes, it is allergic to the skin, and that material is not compatible enough. Major disadvantages with this is the ethical issues which will be coming into picture, because we are going to sacrifice an animal. In case if the material turns to be toxic or carcinogenic, the animal will be sacrificed. Hence, just for testing the material response, biological tissue material response for the implant, for the biomaterials, we can't just sacrifice the species just like that. So the standard regulatory boards would be looking into the news concern based upon the study results of the preliminary test, that is the in vitro test. They will decide whether it can be given for animal test or not, and the species chosen also whether it is of close match with the respect to human tissue or not. If it is not, then they don't give the permission. So they can be difficult to interpret, control, and also expensive and also time consuming, because after implantation we need to wait for the response to elicit and the any sort of reactions onto the skin, onto the functioning of the behavior, onto the behavioral patterns. Is there any change or not? Everything will be analyzed here. The relevance of the test in the in vitro of the material is quite unclear, especially the estimating the appropriateness of animal species to the human. Though we have selected the animal species which is having close match with that of human tissue, yes, it is subjective in behavior which is getting involved in it. If it gives 100% success in the animal test, it need not be necessary that it gives 100% results in the human tissue, since there is a variation with the species levels and all those things. Yes, there is only the assurance which we can, if it is safe for the animal, yes, it is completely safe for the human. Are the reactions going to be changed? Not necessary. There may be a change, may not be a change. It may give the same result, or there may be a variation in it. So in vivo test at the level 3, which is of major interest as it involves human volunteers in the analysis. So for this, we will be using various levels of testing. Like we start from cytotoxicity, which is of major interest for the in vitro. So you're starting to the preliminary study to the entire implantation. Entire analysis would be done on the human volunteer in order to assess its toxicity at the level of cell, then at the level of chronic or acute, like for prolonged application, is there any change or not, or any short-term reactions which is going to take. Then is it going to be toxic at the level of DNA, gene mutation, mutagenic changes are there? Is it going to cause any sensitization, skin irritations, reddishness, allergies, and all those things? Systemic toxicity, like I told you, like once these elements, material implants, they start getting leached, and these particulate material, leached material, tries to enter into the lymphatic and circulatory system. Yes, they tend to change the response on the systemic level, like not only in the host tissue, the surround tissue, and then it will be carried throughout the body, and the infection also spreads throughout the body. So that is at the systemic toxicity level. And if the leached particles are entering into the bloodstream, into the circulatory system, then it is of major concern, because blood is the major medium which supplies nutrients and reaches each and every cell of the human body. So if it is getting contaminated, that contamination will be carried to each and every cell in the human body, and it would be finally resulting in carcinogenicity. Irritation, skin irritations, it may be skin irritation, membrane irritations, or any other inflammation. So finally they will decide whether to implant or the material inside the body or not. Is the material compatible enough to be used as an implantable material for a given application? That will be decided. So usage test, as I told you, based upon the results of in vitro, in vivo animal test, we would be selecting the species. Here also, prior going, if we are not getting any cadavers, or the cadaver is a dead body, few of the analysis can be done in on cadavers also. And if you want to get human volunteers to be involved in it, so live applications of the implantable material will be analyzed by taking into consideration the human volunteers. So prior involving human volunteers, we will be choosing the animals of sophisticated metabolism, like which includes monkeys, donkeys, dogs, and all those things. So as I told you, it may be done in animals or human volunteers. So fidelity with which the test mimics the clinical use is of the material in every regard, including the time, location, and window. So entire complete picture of the tissue material interface with respect to time, with respect to host tissue, with respect to the material properties, everything can be clearly analyzed in this test. So the test material is placed in the situation identical to its how you want it to perform inside the body. Exact the same application will be done in this test. For this reason, usage test in animals employ larger animals such as dogs, monkeys, and humans are used for these usage tests, and such tests are called as clinical trials. So if a human is involved in the usage test, then those tests will be called as clinical trials. Advantages of this test is they are totally relevant and must and should for the commercialization of that product into the market and get it implanted for the patients. So these tests are gold standard and give ultimate answer whether the material is compatible or not. So the final decision on its biocompatibility would be given based upon the results of the usage test. Since it is using animal species and human volunteers, yes, it is quite expensive when compared to the in vitro and the animal test, and it has to be done for longer periods in order to study for the systemic toxicity and involve ethical and often legal concerns, because even humans are also involved. Getting a human volunteer itself is very difficult, and after getting that, having proper ethical permissions to be given by the regulatory boards is also another issue. And once if it is turning into any adverse effect or any undesired complications arise, then it may lead to legal concerns also. Are exceptionally difficult to control and interpret accurately. The statistical analysis of the test are often daunting process. So here these test results are gold standard. Like once they give the level of biocompatibility, yes, that will be there for the record, and then based upon those results only we try to commercialize the material into the market for the fabrication of implantable devices. Even then, since we have subject to subject behavioral changes, so a subject, if it is going to give 100% success in one patient, the same material with respect to same design and same application need not give 100% result in the other patient. So that variability will be there, and considering that element into the consideration, like we need to assess the results and then take the appropriate steps. So that is about the biocompatibility test and the various protocols used for biocompatibility. Thank you.
Like, share and subscribe. Hit the bell icon for more updates.